Mute करो।
Good morning, everybody. We welcome you once again to our workshop. And today we are going to have the technical session 4. In technical session 4 of the workshop, Functional Food, Bioactive Compounds and Phytochemicals for Better Nutrition, in the technical session 4, we have two eminent speakers. Our first speaker is Dr. Vreda Krishnan, Scientist, Department of Biochemistry. And our second speaker of today is Dr. Vinuta T. Gowda, Scientist, Division of Biochemistry, IIRI. After the session, after the lectures, we have a bunch of activities today. We have the hands-on training on AAS and the flash presentation. So without wasting any time, I would like to call upon the convener of the session, Professor Gautam Kumar Pandit, Head of Department of Biochemistry, UBKV, to start the technical session 4. Sir, please.
Good morning, everybody. So today is the third and last day of the 3 days workshop organized by Department of Biochemistry UBKV in collaboration with SPBB. And in this fourth technical session, this is going to be the last technical session of the total program. In this technical session, the session theme of this session is functional foods. And in this session, as Dr. Nanjita Shahana has already mentioned, there will be two speeches by two eminent speakers, both from Division of Biochemistry, IRI. And this session will be chaired by Professor Pradut Kumar Pal, who needs no introduction because all of you have already attended his very beautiful speech on the first day. So I invite Dr. Pradut Kumar Pal to chair the session and take over the charge and continue the program. Thank you very much.
Thank you, Professor Pandit.
Very good morning to all of you. Today, I welcome you on my personal behalf and on behalf of our university to this fourth technical session. We had a very good 2 days of interaction on the subject, and today again we will be deliberating on bioactive compounds, practically functional foods. We have two speakers today. Uh, One from Dr. Veda Krishnan, and second one from Dr. Vinuta Gowda. Basically, functional food, as we all understand, I hope from the deliberation of last 2 days, we all understand that the functional food are the talk of today, and we all depend upon functional food now for, promoting our well-beings. So these two, speaker will be deliberating on two different aspects. One will be deliberating on functional food, their bioavailability, their interaction in the food system, or their functionality as a isolated compound. And the second one is again the functional food is basically formulated by incorporating different kinds of food compound. One of the major ingredient is being the minor millets. So the second lecture will be on the incorporation of minor millet, the problem and the mitigation of those problem. And the minor millet that will be deliberated here is the pearl millet. So basically we will be having a very two interesting session. So for the first talk, uh, will be delivered by Dr. Veda Krishnan. Just to give a short introduction about Dr. Veda Krishnan, she is a scientist of Division of Biochemistry, Indian Agricultural Research Institute, New Delhi. She graduated from Kerala University in the discipline of botany and biotechnology during 2003 and 6, and securing second rank during her undergraduate program. She was awarded University Gold Medal for securing first rank in her postgraduate studies from Cochin University of Science and Technology in 2006-2008. During her PG studies, she was also awarded for Best Master Thesis Award from CUSAT. Besides scientist at IARI, she had also catered as a scientist at Chemical and Biochemical Processing Division, Sirkot, Mumbai. As research fellow at Molecular Immunogenetics Lab, Institute of Genomics and Integrative Biology, Delhi. As a junior research fellow at Division of In Vivo Models and Testing, Sri Chitra Trinul Institute of Medical Science and Technology, Trivandrum. And as a research exchange student at School of Biological Science, Nanyang Technological University, Singapore. She had qualified several national level examinations like ICAR JRF, ICAR SRF, ICAR ARS with fast rank, CSIR NET JRF, GATE in life science, and various other examinations during her studentship. She worked in several high-end laboratories across India and abroad. Her major area of interest are extraction and estimation of various plant compounds through HPLC and LCMS, expression analysis of metabolite synthetic pathogen, and agrobacteria-mediated cotyledonary mode method for efficient soybean transformation through tissue culture. She has engaged herself in more than 5 externally funded projects by various agencies and scientific bodies. She has published more than 50 research papers, 20 popular articles, and several book chapters. She visited many countries like Singapore and Malaysia. She is also a member of several scientific communities of India and abroad. Besides teaching and research, she is also interested in debate, quiz, culture, science, journalism, and intellectual property rights, gaining prestigious recognition from these activities. With this short introduction, I hand over the platform to Dr. Veda Krishnan. Over to you, Dr. Veda.
Thank you, sir. Am I audible?
Yes.
Thank you,
Please.
eminent chair, convener, and coordinators for this wonderful opportunity. Let me share my screen. Host has disabled the participant screen sharing. Can you please enable, sir?
Sure.
Enable it.
Okay. It's still showing disabled.
Just, yes. Now try.
Yeah. Are my slides visible?
Not yet. Yes. Now, yes.
Okay.
Please carry on.
So, a very good morning. It's indeed a proud privilege and pleasure to join this virtual conference, jointly organized by the very prestigious Society for Plant Biochemistry and Biotechnology, IARI, and UBKB. And it's indeed a privilege, and thank you for having me here. In this testing time of COVID-19, let me start my talk with a positive quote. Once Winston Churchill said that never waste a good crisis. Because in this crisis, this theme has a relevance of more than any hour, that the functional food is the need of the hour. And many more. That's why a lot of conferences, webinars, and intense research is germinating in this direction. And today in my talk, I am going to address a very important aspect, that how the food matrix and its composition is important for the bioavailability. So let me start my talk that as I mentioned, that I'm proudly associated with one of the largest national agricultural system in the world, ICAR. As you can see, this is the pan-India presence of ICAR. And I am associated with the most reputed deemed university of ICAR, the Indian Agriculture Research Institute. And here in IAR at Division of Biochemistry, me and my colleagues are working in the frontier areas of stress response and nutritional sustainability and enhancement. I hope you will all agree that over the last 2 decades, there is a dietary transition or evolution that we observed, and because of that, there are more than 60% death globally is occurring mainly because of non-communicable diseases, and which we normally call as lifestyle disorders. And with these non-communicable disorders, the major fraction of 57% is due to chronic vascular disease, type 2 diabetes, obesity, and many more. And what is the national scenario? In national scenario, again, we are walking on thin ice, because on one side, we have a growing percentage of stunted children, and on the other side, we have more than 10% increase in obese and diabetic children. So it's a very important scenario, and we have to address the relevance of nutrition. And because of that relevance, the UN has envisioned a decade action plan. And I remember in the inaugural address, the Honorable Secretary, Dr. Shelley Praveen, mentioned that among the Sustainable Development Goals, more than 10 to 11 goals are directly or indirectly related to the nutrition and well-being. And many more. Our Honorable Prime Minister also initiated many programs like Poshan Abhiyan and Eat Right and so on, to make aware the people aware about the importance of nutrition. And in this domain, to bridge the gap between food and nutritional security, functional foods have great relevance. And these are the sustainable developmental goals that I already mentioned. And these are the different programs that was taken by our Honorable Prime Minister. And this is the talk, a light outline of talk that today I am going to mention. And as I mentioned, we Indians are, we are inculcated with the knowledge of Ayurveda, because we believe that we are what we eat. And along with that, a lot of surveys, the national and international surveys, got to know the information that even though these communicable diseases are multifactorial, 87% is mainly contributed by the diet. And that's why a lot of research is going on functional foods. And it is also endorsed by the human trials, which concern that along with the regular food, we need the functional foods which have key bioactive nutrients. And what are functional foods? The functional foods, which was envisioned in Japan, in 1998 as food for special health use. And these are normal foods, but which has an additional attribute for health. Mainly, it could be an antioxidant potential, it could be an anti-inflammatory potential, it could be many things. And among the functional food, we can classify them into 4 different types. One is the conventional health foods, and the conventional health foods include the whole grains, the pigmented foods, the pigmented cereals, the superfood like soybean, nuts, and so on. We have modified functional foods like now we know that FSSI is interested that we have to fortify our grains and oil and milk with a lot of vitamins and micronutrients. So these come under the modified functional foods. Then we have a special class of functional food which we call them medical functional foods, because which is very special for the medical class people. Like some are lactose intolerant, some have a phenylketonuria problem, some have a gut dysbiosis. So they are a special class of functional food for them. And the most glamorous among this is the food for special dietary use. Because these days, we are hearing about the immune boosters like anything. And along with that, the diabetic friendly foods, the food for weight reduction, and gluten-free cereals. So this is the major market is shared by these kind of functional foods. And that's why the grocery shelves are flooded with these kind of foods, among which low glycemic index foods cover the major market share. From the cereals, we can see the diabetic rice, diabetic flour, and even diabetic honey, and many seed substitutes. And all these are mainly focusing on the concept that it should be low glycemic index. So when we discuss about glycemic index or glycemic response, we can understand that this is a trait that is associated mainly with the carbohydrate, because carbohydrate is mainly contributing towards the sugar spike in our body. And that's why the main dietary carbohydrate, starch, is a molecule of great interest. And in the starch, you can see that the food is ranked based on the starch bioavailability or glucose release in different terms. And glycemic index is one term that we most commonly use to rank our foods. We used to take like low glycemic index foods are good because it range, the index is less than 55. The millets, it can come under the medium glycemic index, but we say that the white rice, which is of high glycemic index, we have to, reduce our intake. So glycemic index is always a ratio that we use to test our food compared to the standard glucose. Then we also have another terms index like glycemic load when we consider the quantity of the food that we are taking, that is the total available carbohydrate. And but even though the most interesting factor is that even though we are taking the same amount of carbohydrate, the glycemic response in our body under the in vivo system is different for different individuals, and also it is different in the individual under different conditions. So that's why in vivo glycemic response is another important terminology. And along with that, another important terminology is glycemic potential, because glycemic potential is when we define for different crops. So what is the glycemic potential of a particular rice variety? We can compare when compared to a millet or any other kind of variety. And most of the research focused on the starch quantity, but this recent research by American Journal of Clinical Nutrition highlighted that along with starch quantity, starch quality is also very important. So what I mean by starch quality is, even though the starch content is high, there is a profile of starch that is present in the total starch based on its digestibility. So based on the digestibility, we can divide starch into rapidly digestible starch, slowly digestible starch, and persistent starch. That is based on the digestivity profile. So the starch quality should be different among the dietary sources that we are taking, and that could be one factor that is associated with the glycemic response. And as I mentioned, starch is one of the most amazing molecule, and I'm really intrigued by the complexity of the starch. And we have to do a lot in this with this wonder molecule, because it is not only a unique dietary component which is giving us the per capita dietary energy, but along with that, it has multitude of industrial applications. It can be used as a stabilizer, a fat replacer. The industry is needing different type of starch sources along with this glycemic index functional foods. And another interesting fact I want to say is the starch also has a role in evolution. That how we evolved from hominids to the current Homo sapiens, that the recent study mentioned that the increase of dietary carbohydrate consumption has a direct role in the brain development. And that's why the starch is a molecule of great interest. And as I mentioned, that when we consume the starch, so this is what happening in our system, that we take starch from different conventional, non-conventional sources. The staple cereals are the main source of starch. And initially the starch get bioaccessible, that it is released from the matrix. Then it gets susceptible for the enzymatic digestion. The amylase, we know that we have two different types of amylases, the salivary amylase and the pancreatic amylases, which has a proportion of 30 to 70% digestibility. And later it is moved to the intestinal glucosidases, which convert it into the final free glucose form. So this is the starch digestion that is happening in our body. And once it is bioaccessible to these enzymes, these free glucose enter into the systemic circulation, and then it is absorbed by the glucose responsive tissue. And then we call the starch as bioavailable. So based on this, the question that intrigued me, that why different starch has different glycemic response. So that was the topic, that is the topic of my research, that how different type of starch from different sources have a different glycemic response. And when we did an intensive literature survey, we found that there are two set of variables. One set is intrinsic parameters, that is within the food, but another set is within the individual. We can say these extrinsic factors are individual centric factors. For example, we know the mastication or the chewing rate, it is directly affected by to the digestibility, because the more you chew, the particle size is reducing, and that is going to be easily digestible. But we cannot control those parameters in our research. Then what is the metabolic rate of a person? That all depends on the whether you have a sedentary lifestyle, whether you have active lifestyle, and what is the activity of the various enzymes in our body. What is the meal time? How regular is your meal? What is the pre-meal time, secondary meal time, and how fast is your food moving? That is the transit time of your food, and what is the quality of the absorption? Because all these are associated or affected by the different types of genes that is present in your body, the glucose transporters and other. There are so many factors. So these extrinsic factors are individual centric, and we need nutrigenomic studies for that. So we focused our research on intrinsic factors, and these factors are specially present within the food. Okay. So today I will be dealing with these intrinsic factors which are controlling the starch digestibility and glycemic response. So the first most common intrinsic factor that we address is the microstructure of your food, or the microstructure of your grain in case of staple cereals. We know that the brown rice is better than polished rice for diabetic people. So that is mainly because of the bran that is present, and the bran and the layers of the microstructure, the epidermal layers and the pericarp, those limit the enzyme penetration. And that's why the starch is less digestible. So the microstructure of a grain or a food has a lot of importance in starch digestibility. Along with that, the molecular structure of starch is also important. So what I mean by molecular structure is, we know the starch is a combination of amylose and amylopectin, but it is not that easy, because the hierarchy of starch, we will discuss that is so complex that different type of crystallinity can form in starch, and that also affects the digestibility. So that's why different sources of starch has different rate of digestibility. Along with that, how much starch is present and what is the starch quality, that how much resistant starch is present, how much slowly digestible starch is present. So the starch quality and quantity also has a role in digestibility. And along with the starch, what is the matrix of your food? Whether phenolics are involved in it, whether lipids are there, whether certain micronutrients are present, what is the presence of fiber. So the presence of all these metric components can also cause molecular crowding that we normally say in biochemistry, which can also limit the enzyme diffusion and as well as enzyme based digestion. And further, more into the dynamics, because the presence of this metric component is one thing, but whether they are dynamic or interacting each other is also very important. Because normally we see that the starch can bind with the lipid and form super complexes, which can further limit the starch digestibility, like in case of resistant starch type 5. So that's why in my research and in my today's presentation, I am going to discuss more on these intrinsic aspects of starch digestibility. So as I mentioned, the microstructure, definitely have a role as a barrier in limiting the starch digestibility. We initiated with light microscopic studies with understanding the structure, but later we understood that scanning electron microscopy is very important because it can help us to understand not only the morphology, the topology, and as well as the ultrastructure of starch granules and its packing and so on. So as you can see here, we can understand the granule morphology. The starch granule differ among the domain, and the smaller the granule, better the digestibility. So that itself endorses that why white rice starch is more digestible compared to a barley starch or an oat starch. So that's why the scanning electron microscopy is a very important technique in starch quality characterization. Along with that, because the surface to volume ratio is very important, and that's why the granule size is important. Along with that, the topology is also very important. Some studies has reported that the presence of certain surface structures like pores or channels also can assist the digestion, because these pores and channels, what it helps is like it helps the enzyme to penetrate inside and the granules start digesting from the inside. We call it as inside out digestion. So this kind of digestion will be easy in those starch varieties or those starch from those crops where these have these kind of structures like pores or channels. So they will be easily digestible. Many more, we also use this technique to understand how the amylose and amylopectin combination cause the starch packaging. So in this recent paper we published, we understood that higher amylose can cause a complex starch packaging, and that packaging is reducing the starch digestibility. Further, to understand the molecular structure, as I mentioned, that starch is not a simple molecule, it is highly complex. And as you can see here, the starch granule, and the starch granule, we normally see a lot of growth rings, and these growth rings are an alternate layers of crystalline and amorphous lamellae. So this crystalline lamellae, you can think it is a very compact structure, and amorphous is a bit loose structure. And these structures are formed by certain structures called blockets, and blockets are made of superhelices, and the superhelices are made of a combination of amylose and amylopectin. When we say that amylose is a linear glucose chain and amylopectin is a branched chain because it has alpha 1,4 and alpha 1,6 linkage. But it is not that easy because the same glucose units in presence of water molecule, it can attain different crystal structure. It can have a square crystalline structure. It can have a hexagonal crystalline structure. As you can see here, when it becomes a square, it becomes an A type of crystallinity. Hexagonal, it becomes a B type crystallinity. And similarly, the branching of amylopectin can also cause a different molecular structure like L type amylopectin, S type amylopectin. So there is a lot of molecular diversity in starch, and that is very important to understand. And based on the previous information, we could gather that in cereals, which are normally considered as easily digestible crops, they mostly have an A-type crystallinity. That is mainly because of the how the crystal is formed in the presence of those water molecules. That is helping the enzyme to digest fast. But in case of tubers, they observed the B type of crystallinity, which is more complex and compact, hexagonal lattice, and which limits a better digestibility. And sometimes in beans, kidney beans, and so on, they have found a combination of these two, and they call them a C type crystallinity. We also observed that a combination of starch and lipid, if it is there in the matrix, there is another type of crystallinity called V-type crystallinity. So this crystallinity causes more resistance towards digestion, and that's why it is considered as resistant starch. So to understand this, we took the assistance of a technique called XRD, that is X-ray diffraction. In X-ray diffraction is mostly where you bombard your sample with the X-ray beams. And as per the Bragg's law, we all have studied that based on the diffraction of these electron upon the sample, we can understand what is the intensity. And from the intensity, we can understand how the crystal is compact and what type of crystallinity is present in the starch. So we also performed such experiments, and we understood that rice or white rice varieties, we screened n number of white rice varieties. Mostly we found they are of A-type crystallinity, while certain pearl millet varieties, certain land races, and as well as pigmented varieties, we observed that they have a B-type crystallinity. So that itself can also, add another information that this type of starch could be a low digestible and low glycemic response. The third intrinsic parameter, what I mentioned, is the metrics, because it is not only the starch, whether the starch is available for digestion, that is also important. So mostly we initiated our experiments in extracting each proximate composition analysis that we do in biochemistry, where we extract and then we quantify. But that is not a right kind of experiment in nutrition study, because I think that is only the tip of the iceberg. For understanding the bioavailability of any nutrient, we have to study it in the food matrix, because in the matrix, that nutrient is going to behave in a very different manner. Their bioaccessibility is different, the bioavailability is different. I'm not going to get into the detail because I know yesterday you had an excellent talk by Dr. Anil Dahuja. So already explained well about the bioavailability, but I want to endorse the point that it is very important to study in the matrix itself, because the food is never the sum of the nutrients. So that is very important. And why it is? Because there are a lot of interactions that is happening among the nutrients in the matrix. It could be an enhancement interaction, it could be a masking interaction, it could be a neutralization interaction, it could be these kind of interactions. And these kind of interactions you cannot study when we isolate the dietary component and we estimate and we characterize. So that's why, for understanding the metrics, we took the assistance of confocal microscopy. As you all know, confocal microscopy, where we, laser optical sectioning of the sample is done by lasers, and that's why we get very good resolution. And with the help of fluorescent tags, we normally tag stars with FITC, protein with rhodamine, lipids with Nile red, and then we can understand how the inherent starch matrix exists. What are the biomolecules that is really present in the matrix in the presence of other dietary nutrients? Along with it, we are also using this technique to understand how the starch is getting gelatinized, because the gelatinization temperature and rate of gelatinization of starch is also different. So once you label the starch molecule with FITC, you can understand that how the starch molecule is also gelatinizing and what is the rate of gelatinization. So this is another technique, because this conference is mainly focusing on different techniques. So I'm giving an emphasis on the techniques that we are doing in our research to understand more on the starch quality aspect. The next, as we mentioned, that the quality of starch is very important. We developed an in-house method where we could understand that what is the profile of these three types of starch, that is rapidly digestible, slowly digestible, and resistant starch. So the fraction of starch that digests within 20 to 30 minutes, we call them as rapidly digestible. The fraction that digests within 120 to 180 minutes, we call them slowly digestible. And there is a fraction that evades the small intestinal digestion, get into the large intestine, where it acts as a prebiotic to nourish our gut microbiome, which is also very important. So that is called as resistant starch. So we profile them in different crops, in rice, in wheat, and in pearl millet. And there's an example what we recently published, where we could observe a superior digestibility. As you can see in the digestogram, a superior digestibility is there for white rice compared to the black and the red rice. And from that, we could profile that how much SDS and RS is present. And here I also want to add another term, that is nutraceutical starch. Nutraceutical starch is a combination of slowly digestible starch and resistant starch. And I think that should be the parameter we should check to, it should be the screening marker when we are looking for starch sources for low glycemic response, because your slowly digestible starch is going to give you an extended glycemic index, and your resistant starch is not giving any much calorie output. So that's why nutraceutical starch can definitely be an index for measuring the starch glycemic response. Next, whatever results that till now for the last 3 years we are working actively in this domain, and whatever we understood about the food metrics, so I have concluded that into few case studies, so that how we evolved from a target nutrient approach in nutrition towards a food metrics approach. So as I mentioned, we also initiated our work. So whenever we get a functional food like a soybean or a pigmented rice, we first identify the key bioactive, we characterize, and then we link to the functionality. So that is what we normally do with the functional food. So in this paper, we have done the same, where we fingerprinted the exotic soybean varieties because they were differentially colored. Then we understood the presence of anthocyanin. We profiled the different types of anthocyanins like cyanidin, thionidin, petunidin, delphinidin, and so on, so that we could understand that how much bioactive is present. Then we linked to its antioxidant potential by function assay, and then we confirmed that these are good for, these have potential applications for chronic vascular disease or diabetes. So these are the initial kind of nutrition studies that we used to do. Then we evolved one step ahead, where we understood that we have to validate the functionality of the bioactive, which is very important, rather than extrapolating that the antioxidant potential is going to have an impact. So for that, we had certain publications where what we did is we, again, with the black soybean, we had this pipeline of work where we use certain cell models, like in vitro cell models, to validate how the cytoprotective nature or how the reactive oxygen scavenging effect is there with the key bioactive. So there we are getting more focused into the role of bioactive towards the functionality. So that is considered as a second evolution in the research that we did at biochemistry. And but still the limitation that we were not addressing the metrics at the metrics level. So there, in this study, a recent study, we understood the complementation in the metrics. Because if we extract two components, it is going to interact in a different form. But in a matrix, like red rice, we did in pigmented rice, we found that the presence of nutraceutical starch and proanthocyanidin, that unique matrix is something that is causing a low glycemic response for red rice. Because the nutraceutical starch, I already mentioned, it has slowly digestible and resistance. So it is giving a low glycemic response. But many more. What is happening to the glucose that is released? The proanthocyanidins present in the red rice, it is again assisting, as you can see, the GLUT transporters, so that it can uptake more glucose into the intracellular condition. And further, the proanthocyanidins are regulating the intracellular glucose homeostasis by regulating the gluconeogenesis and glycogen synthesis. So that's why it is very important to understand how the metrics and the metric components together is playing a role in a functional food. So pigmented rice is one functional food we are dealing with. We are also working with pearl millet varieties, certain land races, which are very important for our indigenous system. And we are addressing these kind of studies in food metric. And the next case study is synergism. Synergism is that when we understood about metric dynamics, as I already mentioned, that dietary nutrients are interacting together. And because of that, there are a lot of interactions between the nutrients. So when there is between two nutrients, we call them binary interaction. When it is between three, we call them a ternary interaction, like a starch, lipid, protein interaction. And when the polyphenols are also involved, we can say it's a quaternary kind of interaction. So these all interactions are going to affect, again, the glycemic response. So for understanding that, we had this particular research recently where we introduced exogenously certain fats, cooking fat, because we have a tradition of adding different cooking oils in our cooking preparations of rice and so on, so that these traditional systems also had certain scientific background which we were not understanding at that particular point. So understanding the metrics helped us to move one step ahead, and we found that adding these oils have a role in reducing the glycemic response, and among which we found that it is not only the crowding, not only the molecular crowding which is limiting the glycemic response, it is also limiting the starch digestibility by altering the starch structure. So that we understood by FTIR, where the vibrational spectroscopy will let us know that how the COH bond or the COC bonds are modulated by the presence of lipid. Based on that, we proposed this novel model of lipid-induced resistance, where we correlated it with the presence of saturated fatty acid, unsaturated fatty acid, and as well as in red rice alone. So the matrix is changed in three conditions. So in alone, it is the natural matrix, but when we are adding lipid exogenously, which is rich in saturated fatty acid can directly bind to the amylose chain in a very compact structure form, as you can see here, and in this A figure, and that's why it leads to a B type of starch crystallinity, and that can cause more RS formation. But when unsaturated fatty acid are more, what we observed is because of the kinged structure of the double bond, contributed by the double bond, more quaternary complexes can be formed. And that's why that is considered as superior in RS generation and as well as lowering the glycemic response. So that is one study that really made us understand that how metric dynamics is happening. And based on the information that we understood, we also found that different starch sources have different amylose chain length. So the degree of polymerization of chain length is also very important. And our knowledge on different chain length and how fatty acid combination and its saturation. So we recently communicated this as a review to understand how starch-lipid interaction can affect the starch glycemic response. So last but not least, currently we are working on pearl millet, because pearl millet is also a wonderful functional food. Because for me, it is more interesting as the matrix also has a lot of phenolics and a lot of lipids, endogenous lipids. So that's why it is more interesting to understand metric interaction studies. And here we understood how masking or inhibition is happening in the matrix, because the presence of polyphenols can inhibit the starch digestibility by digesting certain carbolytic enzymes. So for understanding that, what we did is we helped the assistance of certain molecular docking tools where we identified certain polyphenols like luteolin, acacetin, and certain phenolic acids like salicylic acid or malatoic acid. So these are specifically present in pearl millet, and these have a role in inhibiting the starch digestibility by inhibiting the carbolytic enzymes. So we are moving now further ahead in characterizing these, polyphenols and how this masking is happening in the food matrix. That is the new ventures that we are taken. And but even though understanding the metrics, finally we have to end with bioavailability. And the bioavailability currently, we have a static model where we are doing a compartment-wise digestion. And with this digestion, we are understanding how the starch is digested and how glycemic response, in vitro glycemic response is happening. But being an in vitro method, it has static method, it has its own limitation. Because in static system, there is a fixed ratio of your food to the enzyme. You cannot modulate the pH, you cannot modulate the electrolyte, you cannot do that. So that's why now we also have a collaborator in Agri Canada. So we send our promising samples to them to understand how the starch digestion happens in a dynamic model. So they have a dynamic gut model system for upper intestine and lower intestine, so that we can do the compartment-wise digestion and we can understand how the glycemic response is happening. And very interesting data that they shared, that they also endorsed that red rice have a low glycemic response, but we observed that the two different compartments, that the intestine has duodenum, jejunum, and ileum. So in their study, they found that jejunum has a low glycemic response, but ileum, to our surprise, it was having a better glycemic response. So that shows the complexity of an in vivo system, that it is not easily translatable in vitro condition. But we have to go through these steps. We have to go through in vitro conditions because these in vivo models are very expensive, and we cannot do that for screening purpose. But once we screen the potential sources, we can validate our research in dynamic models. And the research is very important because we have to develop a regulatory landscape for functional foods. Even though we have a lot of hue and cry about functional food, but still the health claims are coming, based on the manufacturer and this thing. So FSSAIs these days, they are also working rigorously in this aspect, because every country has different aspects on the health claims. So we, from the as a scientific, supporters or the researchers, we have to develop stringent data so that that can be assisted in developing a regulatory landscape for functional foods in our country. And along with that, with the future outlook. In the future outlook, because the consumers are, they are so much interested in functional food these days, not only because of the pandemic, because the awareness that happened over the years. And there is a growing of about more than 15% of this kind of special dietary use functional foods. And that's why we have to have intense research, regulation, and as well as consumer outreach. So thanks to my students who very interestingly depict what we do in science in the form of these cartoons, so that the consumer outreach or the public outreach is also very important to our research, because that should be conveyed among the common people. And many more. We also have to get into the more research domains like foodomics, nutrigenomics, and how these functional foods interact with the gut microbiome. Gut microbiome is also a very important aspect, because those are also important component of our nutrition and our well-being. So we also have to take one step ahead. In future, we are planning to how we can perform these kind of studies to understand how the food metrics assist in glycemic response and as well as other attributes. So I hope I'm almost on time, and I would like to take this opportunity to reflect my gratitude to my support poles, my mentors, my team, both at IARA and as well as my international and national collaborators, because this is because of the support of them and the generous funding that was done by ICAR and other funding sources. I am deeply indebted to them. And many more for giving me this opportunity, this prestigious society, well navigated by the most eminent Dr. S.L. Mehta sir and our very own Dr. Shelley Praveen, Secretary. Thank you for all the support for a young scientist like me, for a budding researcher in this wonderful platform. And I would like to thank my students and all who supported me on this venture. And thank you all for the patient listening. Thank you, sir.
Lecture. We enriched a lot this morning. She talked about, she started with non-communicable disease, its impact, and then linked it to the functional food to alleviate this non-communicable disease. She talked about low glycemic index food. She stressed upon the starch quality and the dietary source, how are going to affect the glycemic response in a food. The starch quality characterization is very important, as he said, in determining the digestibility of starch. Integrity of the starch granules and crystallinity is being studied through different techniques, he told, like scanning electron microscopy, XRD, confocal microscopy. She also emphasized on the matrix dynamics and the interaction of macromolecule in modulating glycemic response. She talked about masking effect of different bioactive compounds in giving ultimate response, physiological response. Uh, Nicely, she explained about the in vitro dynamic model to study the viability of different molecules. Really, we were enriched today to know some of these frontier research that are being carried from one of the prestigious laboratory at the division of the biochemistry at IARI. Thank you very much, Dr. Veda. Now the forum, I'll keep it open for maybe 2-3 minutes for few 2-3 quick questions. Can you anyone please raise your hand? Maybe 1 or 2 quick questions. Well, I hope there are no. It has been well understood by all the participants. So thank you, Dr. Veda.
Thank you, sir. Thank you. And I'm free to interact even the offline session also. So students are welcome. Thank you.
Okay, thank you. So we move on to the next topic today. We have another invited lecture from Dr. Vinuta Gowda. She will be delivering on efficient technologies to limit the rancidity in nutri-rich pearl millet and method to improve its dough quality. I take this opportunity to introduce to the participant that Dr. Vinuta Gowda is working in the capacity of scientist in the Division of Biochemistry, Indian Agricultural Research Institute at New Delhi. She was graduated from University of the Agriculture Sciences, Bangalore, and also did his master degree from the same university. After that, she moved to IARI New Delhi for doctoral degree program. She was qualified several national level examinations like ICAR JRF, ICAR SRF, ICAR ARS, etc. Several awards like IARI Merit Medal for outstanding academic performance in PhD program. She was also awarded DVT Global Bio India 2020 Best Poster Award for development of efficient protocol for natural vitamin extraction from soy meal. Startup research grant for young scientist in the category of the life science by DST. She was also awarded Young Scientist Award by the Society for Biotic and Environmental Science, named to her credit. She had guided 2 MSc students so far and patented 5 technologies till date. She has published more than 50 research papers and more than 10 books in different national and international journal of repute, and several popular articles also published by her. She is also a fellow of several scientific bodies in India. Now I invite Dr. Gora to deliver her talk, and we'll be listening from her on efficient technologies to limit rancidity in nutri-rich pearl millet and method to improve its dough quality. Over to you, Dr. Gora.
Yes, thank you so much, sir, respected Professor Pradyut Kumar Paul, today's chair of the session, and convener Professor Gautam, and my classmate and my dear friend Dr. Somnath, who is my friend and who me and Somnath we both studied together here in IRI campus. Thank you, Somnath, for having me here, and also I want to thank Nandita. Nandita is also, she's my junior. Myself and Nandita, we both worked in the same lab and we have shared the same bench space, and under the able guidance of Dr. Shelly Praveen. And I want to thank all the members of the organizing committee, which is jointly organized by Society for Plant Biochemistry and Biotechnology, IRA New Delhi, and Division of Biochemistry, UBKV. And I welcome you all to my talk. Let me share the screen. Can you able to see my slides?
Sure, yes.
So I welcome you all to my talk on a novel processing technique to reduce rancidity and to improve the dough quality of pearl millet flour and its effect on nutritional quality and functionality. As you all know that the pearl millet is a nutri-rich cereal, and it holds goodness of nutrition like high protein content, high PUFA, high iron content, zinc, essential amino acid content, and dietary fiber in it. Despite all these nutritional benefits, the pearl millet is not popular among consumers and it is not acceptable by food industries. There are two major reasons which hinders the commercialization of this pearl millet and its pearl millet derived products is the rancidity. That is, whenever the pearl millet flour is kept for storage, when it is stored more than 10 days, it leads to a rancid development. And also there is a problem of poor dough quality. That is, the dough quality is very, very important for the baking products and for the chapati making quality that we all know that the excellent dough quality which is present only in the wheat. And these two problems which hinders its commercialization. So let us see the biochemistry of rancidity development in pearl millet. We all know that the lipid content is very, very high in the pearl millet flour, that is 5 to 7%, and the rancidity which takes two pathways, that is enzymatic and non-enzymatic pathway. So if we see the enzymatic pathway, there are 4 different enzymes which are involved in the rancidity development in pearl millet. That is initially the lipase enzyme, which hydrolyzes the lipid into a free fatty acid content, and this free fatty acid content further they are oxidized by a lipooxygenase enzyme, and it gives to a lipid hydroperoxides, and these are called as a primary oxidative products. And these primary oxidative products further they are oxidized by peroxidase and polyphenol oxidase, and this further oxidizes to give you a secondary oxidative products like aldehydes, ketone, MDA, etc. And then it also leads to a free radicals, and this free radicals further it acts on free fatty acids again, and it will lead to a generation of primary oxidative products. And if you see the non-enzymatic oxidative pathway, it is like hydroperoxides, again they are further oxidized non-enzymatically to give you a secondary oxidative products and leading to a off flavor formation and rancidity development in the pearl millet flour. To address this problem, we have developed a technology where it has two components. One of the component is a hydro treatment or a soaking, and other component is a thermal treatment. And the hydro treatment or a soaking is a common step which is being applied in this particular technology in order to activate the enzymes present in the millet. And we all know that the water content in the grain, it activates the enzymes. So when all the enzymes it will get activated, further it was subjected to a thermal treatment, that is hydrothermal treatment, which is also called as a heat moisture treatment, in the sense that hydrothermal treatment which is given at a high temperature for 5 minutes, and further then it was subjected to a near infrared rays treatment, and the near infrared rays range which is being used here is 0.7 to 1 micrometer, and this particular infrared rays treatment was given at the temperature of 40 degrees Celsius for 5 minutes, and later on. And these three major steps which are involved in this particular technology is basically has a different role. The first step in this technology is the hydro treatment, which actually activate all the enzymes, where we could see the acceleration of the enzymes like lipase and PPO by 8% and 19%. However, the LOX and POX activity has been decreased because of the temperature condition, which is not conducive to the those particular enzymes to get activated. And in the second step, where it is an important step of this technology, is the hydrothermal treatment, which has been given at 100 degrees for 5 minutes, where the heat moisture treatment, which the water in the form of a steam, it will convert and then it will introduced into the wet flour material, and then that particular treated flour will again subjected to a near infrared rays treatment. Both it serves the both purpose, like a reduction of enzyme activity and the reduction of rancidity. Another one is to quick drying method. So in future, anybody can use this near infrared rays treatment not only to reduce the rancidity or reduce the activity of the enzyme, but also can be used as a quick drying method in order to bring down the moisture content to a desirable level. And this whole technology now it is being applied for the patent, and this particular work has been taken under a prestigious program of ICAR Niche Area of Excellence program, which is currently being led by Dr. Shalli Praveen. So later on, once the hydrothermal, this particular thermal treatment was optimized, now this optimization we have done for the 5 minutes based on the no color change in the flour, because we have to consider all into account, not only the reduction in the rancidity part, but also we have to take into account the how the appearance of the flour should not get changed. So based on that, we have kept the time limit for the 5 minutes, and later on this thermally treated fresh flour was further subjected to analyze the enzyme activities, nutritional compositional changes, and molecular structural changes in starch and protein has also been analyzed. And later on, this thermally treated flour was stored for 3 months or 90 days at room temperature, and again it was analyzed for hydrolytic and oxidative rancidity. So if we see the lipase activity in the combined thermally treated flour, there was a significant reduction in the lipase activity was observed, that is 48%, and the LOX activity has also been reduced to an 85%. And if you see the peroxidase activity, there is 98% reduction in the peroxidase activity, and there was almost there is a nil activity of polyphenol oxidase was observed after thermal treatment. And later on, in the stored flour, we also have analyzed the hydrolytic and oxidative rancidity. So the hydrolytic rancidity was observed that the 90% reduction in the hydrolytic rancidity at the 60th day of storage at room temperature, and again the 68% reduction in the hydrolytic rancidity was observed at the 90th day of the flour which has been stored. And later on, the oxidative rancidity was also being analyzed, and it shows that the 98% reduction in the oxidative rancidity at the 60th day of storage, and 66% reduction in the oxidative rancidity at the 90th day of storage. That means it shows that this particular technology, that is the thermal treatment combined with the hydro treatment, which has been like, I could not able to see my slide.
Share the screen again.
Okay, can you see now?
Yes. Yes. Yes.
This particular technology now, it has been proved that it is highly effective in reducing the rancidity even if it is stored at room temperature for 90 days. However, we have tested that initially for the only one popular varieties. Later on, we have taken the several varieties which are called as a zonal varieties of pearl millet, and again we could able to see the similar trend in the reduction of rancidity when it is stored for the 90 days. That means it is irrespective of the varieties, that particular technology is highly efficient in reducing the rancidity. And based on this, we have developed a rancidity matrix. And this rancidity matrix, which is based on the free fatty acid content and the total hydroperoxide values. And here, whatever you are seeing, the colors, the red color, which denotes highly rancid region, and the orange color, which denotes the medium rancid region, and the green color, which denotes the low rancid region. And after subjecting the thermal treatment to all these varieties, we could able to see there is a shift in the rancidity matrix from orange and red towards to a green zone. So this entire experiment, which concludes that this particular technology is highly efficient to reduce the rancidity in the pearl millet flour. And this has been now applied for patent. And after successfully established or developed this particular technology, we just wanted to know whether, though the reduction of rancidity has been successfully, it has been addressed, now we have to see whether the nutritional composition of the millet flour has been changed or not. And similarly, we need to see that whether any structural changes in the protein and starch which has happened. So though for that, we have also analyzed the nutritional compositional changes which has not been shown here, and where we could able to see no change in the total protein content. However, there is a small change in the soluble protein content, it was decreased. And similarly, there was the composition of the starch has been changed. We could able to see the structural changes in the FTIR, the Fourier transfer infrared spectroscopy. We could able to see there is a structural changes has happened both in starch and protein. So if you see the wave number or the spectra of 1047, which denotes the ordered crystallinity on the starch granule surface, and 1028, which denotes an amorphous region on the starch granules, and this ratio of 1047 to 1028, which denotes the ordered crystallinity of the starch to amorphous region, and the 1028 to 995 ratio, which denotes the amorphous region of the starch to a crystallinity. So if you see that after combined thermal treated flour, there is a significant decrease in the ratio of 1047 to 1028. It denotes that, or it is implying that there is a disruption of crystallinity in the starch granule surface, and that it enabled the sensitivity of the starch molecule to a digestive enzyme. And it is also that is highly it is further so that it enabled the easy attack of amylolytic enzymes in the starch, and that was being again analyzed by increased starch digestibility in the thermal treated flour. And this increased digestibility is in a way that we could see that there is an improved digestibility has happened after after thermal treatment. And not only we could able to see there is a disruption in the crystallinity of the starch granule surface, but we could also able to see there is a fragmentation of the starch granules into a smaller size that has also been captured under confocal microscope. And later on, we could also see there is a structural changes in the protein content like alpha helical changes, beta sheets, and random coil. There was a change, but this change were insignificant. They are not significantly, much change has been occurred as compared to a native flour. And the protein digestibility has also been analyzed, and the in vitro protein digestibility, there was a decrease in the protein digestibility, not to a significant level. The 1% decrease in the protein digestibility was observed, and the degree of hydrolysis has also been analyzed, and there was a non-significant change in the degree of hydrolysis in combined thermally treated flour. So this entire methodology, this entire experiment, it proves that yes, there is we could able to successfully reduce the rancidity in the pearl millet flour when it is stored in the room temperature. But apart from that, we could able to improve the starch digestibility in the pearl millet flour, and there was no such changes which are occurred in the protein digestibility, or the protein structural changes which are occurred are non-significant. And later on, we will move to how to improve the dough quality of the pearl millet flour, and this is another second most important problem in this particular area. And for this, we have developed a cost-effective methodology to extract the vital wheat gluten from wheat and its regeneration in pearl millet flour. We all know that the vital wheat gluten is an important commodity, and it is currently we are importing all the vital wheat gluten from China, Australia, Spain, and Germany to a value of $44 million. And all the imported vital wheat gluten is currently being used in the bakery products. That is more than 55% of imported gluten. It is being used in the bakery products, and rest in the sports nutrition, extruded snacks, and also to some extent in the flour fortification. So this shows that that currently that India, it is not taking a momentum to solve this problem or to extract the vital wheat gluten because of several reasons. One of the reason is that the low technological development, and we all know that the India is a leading producer of wheat. It is the second largest producer of wheat in the world, and we have, if you see the surplus of the wheat, it is present in the India's food basket, but the storage loss is also 8 to 10%. It has been recorded. In spite of that, we are importing the vital wheat gluten to a larger value. And this to address this problem, we have taken up this particular the problem to solve this problem. And before that, I just want you all know that what is vital wheat gluten. It is an extracted form of a gluten which has the ability to form a network upon hydrating and mixing with the wheat flour to give a typical viscoelastic behavior to dough. It is called as a regeneration of vital wheat gluten, and it is as seen naturally. We will see this particular viscoelastic form of the gluten in the wheat dough, and this gluten is a protein which is made up of a glutenin and gliadin, and upon hydration these two will combine together to give a viscoelasticity to the dough. And this particular problem we have taken up to extract the vital wheat gluten, the gluten in the form of its fully vital form, in the sense that it should retain its the viscoelasticity in it. So for that, several patents have already been available, and they are for the gluten extraction. So one of the US patent, it says that they have used a drying temperature of 155 degree, and they have dried in a ring dryer. And but then the problem in this particular patent was that there was an initiation of rancidity was observed, and since they have used in high temperature, which promotes the disintegration of the gluten, and this disintegration in turn means it is a denaturation of the protein, which supposed to be supposed to be like it should not be a denatured condition. In another US patent, they have done the extracted vital wheat gluten, though they have not observed any denaturation of the protein, since they have used an hydrophobic phase and they have dried under the reduced pressure. And the limitation is that the high capital expenditure and having about in order to remove the 50% carrier oil, and also removing this oil component would require an additional expensive steps. The other US patent, they have used an alkaline water solvent phase, and they have redispersed the viscoelastic mass in a suitable solvent, and further they have purified by ultrafiltration. Again, the problem here is that the complex steps, it is in the process of the separation of starch and gluten, it was involved, and the extra equipment it was needed for the ultrafiltration, and the solvent recovery system would require again a large capital expenditure. In other US patent, they have used a spray dryer, where the problem here is that there was deficient in the good baking quality. And several other patents also have been patented for the enzymatic method to separate starch and gluten. And in this method, in the enzymatic method also, the people have seen there is the chances of losing of gluten is very, very high. Considering all this into account, that the presently we have taken a due care to address all these problems, and we have invented a easy and it's a simple, easy, and cost-effective method to extract the vital wheat gluten with the different steps. And one of the USP of this particular methodology is the drying method. That is the drying temperature what we have used. Different drying methods we have used, and in this different drying methods like high temperature we have used the near infrared rays method, and also we have combination of these two and combination of low temperature and near infrared rays treatment, and again we have used the temperature between 40 to 50 degree. So all this after considering all these drying methods, we could able to then we have analyzed the functional characteristics of vital wheat gluten, and the functional characteristics was analyzed basically by considering two important parameters. So one of the parameter is the water absorption capacity, and another parameter is the elasticity or gluten extensibility. And this particular, the water absorption capacity was significantly higher in case of the way the temperature was used between 40 to 50, and again it was compared with the commercially available gluten, and it is stand apart by having higher water absorption capacity. And this, after that, we also have analyzed the elasticity of the gluten, and where we could able to see a better gluten extensibility of the gluten which has been extracted using this technology as compared to a commercial gluten. And later on, once we have successfully established this particular protocol and the technology to obtain a gluten product fully in its vital form or with the higher viscoelasticity, later on we have moved into improve the dough quality in the pearl millet flour by reconstituting this particular vital wheat gluten in a different proportion in pearl millet flour. And again, the dough quality parameters were analyzed. Several dough quality parameters like water absorption, dough development time, dough stability, mixing tolerance index, and dough consistency has all been analyzed using farinograph, and we could able to see the good dough quality parameters in case of 7% reconstituted vital wheat gluten in pearl millet flour, which was on par with the wheat dough. Again, other functional characteristics of the dough, like a texture properties of the dough, were also been analyzed, like hardness, adhesiveness, cohesiveness, and springiness using texture analyzer. And here also we could able to see a better cohesiveness of the pearl millet dough, which has been reconstituted with 7% VWG, and it was on par with the wheat dough. And again, the dough extensibility is another important parameter we need to address to improve the dough quality. And this dough extensibility was analyzed using Kiefer dough rig method using texture analyzer, and we could able to see a better dough extensibility of the pearl millet dough reconstituted with 7% as well as 10%, which was on par with the wheat dough. And another important property for the dough is the viscoelastic property. And this viscoelastic behavior has also been studied. The linear viscoelasticity region of the different dough samples was studied by a means of a stress sweep protocol in comparison to wheat dough. Similar trend of linearity over the entire frequency range for storage and loss modulus. The storage modulus, which indicates the elasticity, and the loss modulus, which indicates the viscosity, it has been observed in the 7% VWG reconstituted pearl millet flour, which is lacking the accentuated inflection points that could indicate the breakage of sample structure. And all this, the dough quality, it says that the 7% VWG reconstituted pearl millet dough has shown a better dough quality, which was on par with the wheat dough. But then again, we need to also see the tensile strength of the tortillas, which are prepared from this dough. And we could also see that the better tensile strength in the tortillas, which are prepared from the 7% VWG pearl millet dough, it is on par with the wheat tortillas. And this entire experiment, which concludes that yes, we can improve the dough quality of the pearl millet flour, provided we are using the gluten in its complete vital form and it is having a high viscoelasticity. And the potential of the technology, if you see, it is a huge. And India, being the second largest producer of wheat in the world, has cutting edge advantage in export of wheat byproducts like vital wheat gluten at premium price by adoption of this technology at upscaling level. The technology or the product which is developed has the potential to move as a sustainable alternative to wheat in many baked and cooked preparation as a new staple food, owing to its easy availability throughout local markets, supermarkets, and e-commerce sites. This product not only can positively impact these markets but also has the potential to address nutritional challenges, as the hidden hunger and malnutrition is associated with a huge investment from the Indian government, with the estimated economic cost of 0.2 to 0.8 to 2.5% of the total GDP, which is equivalent to 15 to 46 billion US dollars. So this particular technology now, and it is under commercialization, and the companies have came forward to commercialize this product with us, and MOU is under process. And I am very much thankful to our leader, Dr. Shali Praveen. Under her able guidance, we could able to make out all these technologies is coming into a limelight. And also, I want to thank all my colleagues who have helped in this. They are involved in this particular project. Dr. Suneha Goswami, she is a scientist from this division, and Dr. Shalini Gaurudra, she is a scientist in division of food science and post-harvest technology. And also I wanted to thank all my team members, those who are involved in this particular project. And nevertheless, I want to thank our respected director, who has really encouraged our division to take up this kind of work. Thank you all. And I want to thank all the organizers of this workshop, Dr. Somnath, Nandita, and I want to thank our President, Dr. S.L. Mehta, sir. And also I want to thank all Vice Chancellor of UBKV to giving me this opportunity to present my this small work here. Thank you all.
Thank you, Dr. Vinuta. It's pleasant hearing you. Uh, Maybe you can have some question. Okay, Dr. Vinuta has explained, the nutritional quality of palm millet, and this, very important millet. We are not able to use because of few problems. And she nicely, explained how these two problems they have taken up to resolve and come up with commercial technologies. The first she was talking about hydro treatment, hydrothermal treatment, and NIR treatment to elevate the problem of rancidity in palm millet. And secondly, very important aspect of it is that the viscoelastic property of the dough from palm millet, how it can be enhanced by incorporating something like the vital wheat gluten. It was very nice and informative talk. And uh, okay, pigmentation. Yeah, there are some question I am finding here. One I have already seen from maybe Mr. Divan about what is farinograph test. This question was asked in the chat box. And one more thing, how to overcome the pigmentation in palm millet flour. Madam, can you please?
Okay. The farinograph is a kind of an instrument. That instrumentation part I have not explained. It's a very expensive instrument, around 50 to 75 lakhs. And this farinograph is basically just being used to analyze the dough quality. Like what is the dough quality parameters? Like water absorption capacity should be very, very good. That means it should have an increased water absorption capacity so that it will give a pliable, the quality to a dough. And it should, the stability of the dough should be very, very important. That in the sense, if you keep it for a long time dough without making a tortillas or a chapati, it should maintain that stability with the same viscoelasticity. That is very, very important, and it should not lose its elasticity. And another one is mixing tolerance index. It is something like that. It is a tolerance level, in the sense how much ever the force you put, there should not be any breakage in the dough. These are all for most important qualities in the dough one should take into account before analyzing any dough quality of the any kind of dough. And this is globally being used, this particular farinograph test, to analyze the dough quality.
Yeah, another question is how to overcome the pigmentation in palm millet.
Yes, pigmentation, in the sense the pigmentation is not there in all the pigmentation. Are you saying about the gray color in the pearl millet? That we can't do anything. Only thing is that IRI has developed one white variety. In the sense, in the dough, the flour which is prepared from that particular variety, it doesn't have that gray color. It looks like a white color as good as wheat. So that kind of wheat varieties we can I mean pearl millet varieties we can use to again improve the dough quality. Otherwise, right now there is no such technology it is available in order to remove the pigmentation or the gray color in the pearl millet.
Is there any bleaching treatment available for this? No.
Maybe it is available, but then again it is how it affects the nutritional quality and how good it is for the taste should not get changed. That is also one more important component we have to take into consideration.
Uh, okay.
Dr.
So,
Vinita, I have one question, if you
Yeah,
don't mind, if
yeah,
I can ask, we have time. Thanks, and I do congratulate for that the great work, and I do appreciate the team led by Dr. Shelley. I had seen her YouTube video about this also,
okay.
and I really appreciate your work. Congratulations. One thing I just want to ask: then when we are giving this treatment, we are giving NIR, then what is the I think I want to ask that price about the price thing. But if we are treating, because we are treating the flour, you're saying, na?
Yeah,
Then definitely the bajra flour that is available in the market, when we do this treatment, what will be the change in price, and won't that be costlier? The availability will be to
yeah,
people like us, okay, we can afford that, but how can we ensure that to common people who are going to buy this, they will not be much affected by this cost factor, because this will lead to, I think, cost enhancement, I think.
yeah, certainly. If the dough quality is being improved in the pearl millet, certainly the price level will go high. It's no
Yeah.
doubt in it. But it's not that as much as high that one cannot afford it. For example, if you are giving around 20-30 rupees per kg wheat dough, maybe
Mm.
the pearl millet dough will cost around 55 to 60 rupees per kg. So that is how because the basal price of the pearl millet is 40 or 45 rupees. It is not low. If the policy intervention will be there, then
Mhm.
it can be reduced to a greater level. Otherwise,
Yeah.
the cost involvement in the technology itself per se is very, very low. But the commodity cost itself is higher than the wheat.
Yeah, it's higher, and in colder region, in cold season, we generally prefer to have this bajra because it gives immunity and strength in winter season. That's why I was asking, because if it's a big family, then definitely 15 rupees per kg cost, if it is going higher,
Yeah.
then definitely, even for myself, I won't say that because my family is small, but if for bigger family, if we go for that, definitely it will make a difference, I think so.
Yeah.
So
But
yeah, policy intervention needs to be taken care of.
the technology wise, it is around zero point 0.3 to zero point 0.5 paisa per gram of vital wheat gluten. We could able to work it out till now. And that's how. But in the larger quantity, if somebody will take up this entrepreneurs, if they'll take up to a larger level of production, definitely the price will again go down. Because how much larger amount we will do, that much cost involvement will be low.
And as you have said that this can be stored for 3 months, that will also help. Definitely,
Yeah.
that will help. People
Yeah.
will store. Even myself, I'll prefer to buy that. Thank you,
It
sir.
is already then by few days it will be available in the markets because the commercialization is under the process now. It is almost the filing process
Okay,
has been
I'll be, I think, amongst the first buyers. Thank you so much. Thank you, Dr.
completed.
Madam, one more question in the chat box. It is about, is it possible to mix the coconut flour to palm millet flour to overcome the pigmentation?
It's a nice question. Whoever has written, just to overcome the pigmentation, we can do it again. The coconut flour is lead to rancidity.
Yes,
That
yes.
again we cannot keep it for long time. Otherwise, for the short, storage time, yes, we can use. That's a beautiful idea.
Okay, thank you, ma'am. Thank you for the nice response. I once again congratulate you for taking up these two great problem of utilizing palm millet as a source of nutritious food. I thank you very much for your nice deliberation on the topic. Now I hand over the session to the convener of the session, Dr. Gautam Pandit, to conclude this session.
So, thank you, Professor Pal. So we have gone through very interesting two lectures in this morning, and I hope all the participants have gained a lot, not only from these two sessions to speeches. We have already gone through a number of speeches during this 3 days program. So I hope that participants, and particularly the young scientists or faculties, some of them will be encouraged to take up some research works in this field of functional food or nutraceuticals, as because I think out of nearly 3.91 lakh plant species, only a few thousand have till now been explored systematically to search for bioactive compounds. So I think if we devote our time and effort to find newer molecules which may be bioactive in different ways, maybe insecticidally active, maybe nutritionally phytochemical or nutraceutical type, this type of different types of compounds may be available. And because a number of in our nature, Mother Nature has stored a large number of compounds or different remedies of different diseases and problems. So we have to explore those. So what I think that if we search systematically, we can go for or we can get a number of compounds from all these. And I hope that many of our new faculties and young faculties, researchers will take up programs in these fields, and we shall get a number of good molecules from nature. So with this hope, I want to conclude this session, and I thank everybody. And I am a little saddened to hear that we shall not be able to hear any more this type of lectures now on. So hope we shall be able to meet again in future in this way. Uh, so there will be much more programs coming. So with this, I thank everybody for their active participation, and I welcome Dr. Nagesh to take up the charge of the session. Once again.
Okay, so good morning, all of you once again. Today is the concluding day and we have various activities going on. There was an excellent morning session, 2 power pack performances, 2 power pack ladies, beautiful ladies. So excellent talks. I hope all of you have enjoyed. Now moving ahead, there will be the third hands-on training today. And this will be on hands-on training today will be on atomic absorption spectrophotometer technique. And just like the previous 2 days, we have prepared a short video for all of you to have a look on the instrument. And after that, we will be taking few of your queries if you have any. And today I am accompanied by Dr. Prithusayok Mondol, Assistant Professor of Department of Biochemistry, and he will be with us for the demonstration and this question answering session of atomic absorption spectrophotometer technique. So are we ready with the video? Yes. Yes. So let us first see the video and then we'll be coming back. Share করেছো screen? Yes. Yes.
Dear participants, we are facing a little trouble in network. So please bear with us for a few seconds. I think it will be resolved. Ma'am, sir, we are starting the lecture. I mean, practical session again. We shall start the session from the beginning again once the problem is resolved.